welcome to AMP with Dr. H, and I'm going to be talking a little bit about the neuromuscular junction, right? This is all about how do we send a signal from our brain to make skeletal muscles contract and relax. But the neuromuscular junction is just one small part of the piece of the puzzle that causes our muscles to contract. We want to focus on it first before going on to the next step. So let's think about what happens at the neuromuscular junction. This is the junction between the nervous system and the muscle. So what we have projecting down onto all the skeletal muscles of the body, well, we have axons that are coming from the brain. These are all part of the somatic nervous system. And they're going to branch out, and at the end of each one of those axons are these little trees, right? Kind of almost tree branch thing. And at the end of each one of those is a slight, small enlargement. This is the terminal bouton. A lot of people call it other names. Terminal nerve endings is one of the main ones, really popular out there. But I like terminal bouton because it's so much more fun to say bouton than it is to say terminal nerve ending. So if we take and we draw this bouton, what we can do is we can say we've got a cord coming in to an enlarged, almost sac-like thing. Okay. And inside of this terminal bouton, we're going to have vesicles. And remember, vesicles are in essence little phospholipid bubbles that are made way up here in the neuron. Okay? The proteins that are made in the neuron, they get packaged into vesicles in the Golgi apparatus. So each one of these vesicles is going to be filled with proteins, and if those proteins are coming from the nervous system, we call them neurotransmitters. And the specific neurotransmitters that are released to help cause excitation of our skeletal muscles are called acetylcholine. So these right here are all going to be vesicles filled with acetylcholine, and we abbreviate acetylcholine, capital A, capital C, lowercase h. Let's draw on the next piece of the puzzle. And then we'll come back and put it all together. This bouton here is going to come into contact with the muscle. It's going to come into contact with the muscle fiber. And the surface of the muscle fiber is made up of a modified plasma membrane that's called the sarcolemma. And the sarcolemma, well, we'll come back to what's going to happen on the sarcolemma itself. But where the sarcolemma comes into contact with the terminal bouton, that sarcolemma modifies itself. And it's going to create in all of these pit-like structures. These are going to help catch neurotransmitters when they're released. And this is called the motor endplate. This is the terminal bouton. I forgot to label it. Let's go back and label this thing. Okay, this here is going to be the bouton, the terminal bouton. There are a couple more things that we need to make sure that we label. First, we need to make sure that we know that there are lots and lots of transporters in the human body, lots of proteins that span this plasma membrane. And here on the boutons, we have voltage-gated Vg calcium channels, and we have lots of them. So what is the stimulus that opens a voltage-gated channel? Remember, this is a channel that's got a gate on it, and it needs a stimulus to open it. In this case, that stimulus is voltage, electricity, potential. What can travel through these channels? Calcium. Where is calcium normally higher? Outside of the cell or inside of the cell? Calcium's normally highest concentration outside of the cell. There's lots of calcium out here. So when we put something in brackets, remember that means concentration. So there's an increase in the calcium concentration in the ECF. Why? Because calcium's constantly getting pumped out of cells. It's constantly getting pumped out of the bouton, for instance. Pumps, right, run by ATP are constantly driving things from an area of low concentration to an area of high concentration. So we're going to go ahead and draw in a little pink pump here. And this little pink pump is a calcium pump. Calcium pump uses ATP to move calcium from low to high. And it's constantly pumping calcium out 24/7. Every microsecond of every day, that calcium's getting pumped out, pumped out, pumped out, pumped out, pumped out. And only when these channels open can calcium go through these voltage-gated calcium channels. So what is this pump doing? It's creating a concentration gradient that when the channel opens, now everything in the universe is trying to go from an area of high to low. So calcium is going to move from the ECF into that bouton. What else do we need to draw into our puzzle down here? We need to draw on the motor end plate, in these invaginations of the motor end plate, we have a ton of little receptors that hang out down here. And these are receptors that bind acetylcholine. They have a specific name. There's lots of acetylcholine receptors in the body, and the ones that are at the neuromuscular junction are called Nicotinic receptors. These here also bind nicotine. That's why they're named nicotinic receptors. But their primary role is to bind acetylcholine, and it takes 2 acetylcholine to activate a nicotinic receptor. These nicotinic receptors are known as nonspecific monovalent cation channels. Whew, that's a mouthful to say the least, but it's not too bad if you actually break it apart by the words. Nonspecific, meaning it doesn't have a specific ion that's going to travel through it. Mono, 1. Valence, this has to do with charge, electrons, valence shells. 1 charge, cations, positive charges. So these are nonspecific monovalent cation channels. In other words, they allow any ion through that has a single positive charge, the entire first row of the periodic table. Major 2 that are impactful in the human body are sodium and potassium, but these can also let things through like lithium, for instance. Okay? But we're going to focus in on sodium and potassium. So if we zoom in on that plasma membrane, now we got a phospholipid bilayer here. If we zoom in on that plasma membrane and we looked at a nicotinic channel, this is going to be a channel with a hole through it and a gate. It's just a really simplified version of it. On the end of either side of these, by the way, I always draw this ECF, ICF, but on the end of these are binding sites for acetylcholine. So when acetylcholine is released from the synaptic cleft, that acetylcholine is going to filter down by diffusion. I shouldn't have said filter. It's going to diffuse down from areas of high concentration to low, and it's going to come across these binding sites on the nicotinic receptors and bind to it, which now opens up this gate. And when this gate opens, what can pass through it? Sodium and potassium. But where's sodium higher? Outside the cell, because of the sodium-potassium ATP pumps, always moving sodium out of the cell. We got lots and lots of sodium out here. And where's potassium higher? Inside of the cell, so we have lots and lots of potassium in here. Doesn't mean there's no sodium in here. It doesn't mean there's no potassium out here. It just means there's a gradient. So when the nicotinic channel opens, sodium rushes in and potassium leaves. So you might be asking yourself, If sodium enters and potassium leaves, is there any change in the membrane potential? Is there any change in electricity at all? And the answer is, heck yeah, there is. The reason is because what do we have more of in the body? Do we have more sodium or do we have more potassium, or are they equal to one another? Holy moly, is there a lot more sodium in your body than potassium. Think about your diet, sodium-rich diets. You have a multiple-fold increase in the amount of sodium in your body versus the amount of potassium in your body. Therefore, it has a larger concentration gradient. The steep hill, right? We're moving something from an area high to low. The steeper the hill, the faster it rolls. So the more sodium there is, the higher the gradient, the faster diffusion can occur, and sodium rushes in. So we have a net total sodium movement into the cell, a net sodium influx. And what that's going to do is, as that sodium pours into this motor end plate, it's going to end up causing the motor end plate to become positively charged. Why? Because what charge is coming through with sodium? Sodium is a positively charged cation, so the inside of that cell membrane becomes more positively charged. And when that positive charge reaches the sarcolemma again, well, what don't you see on the sarcolemma? You don't see nicotinic receptors. What you see out here on the sarcolemma are voltage-gated sodium channels and voltage-gated potassium channels. And if you do a little bit of research, I don't have time to discuss them in this video, but voltage-gated sodium channels, of course, they're going to be activated by voltage. There's our voltage right there. There's the positive charge that reaches them, opens them up. What can go through these gated channels? Sodium. So sodium's higher outside the cell, and now we rush even more sodium into the muscle, and it causes an action potential. It causes a new electrical switch to be turned on, and electricity is going to rush across this sarcolemma. In addition, there are voltage-gated potassium channels. And again, you could look these up. But now when these open, potassium's higher inside the cell than outside the cell, so potassium rushes out and it's going to repolarize the cell, putting it back to resting membrane potential, putting it back to homeostasis. And when you look at the sarcolemma, there are extensions of the sarcolemma that dive down into the muscle fiber. That's called the T-tubules. What's going to end up happening is that this new action potential that is created is going to end up racing across the sarcolemma, dive down into the T-tubules, and it's going to activate some stuff inside of that muscle fiber. That's not part of the neuromuscular junction, though. So let's focus on that. Steps. The first thing that's going to happen to start to activate the neuromuscular junction is an action potential is going to be traveling. My horrible lightning bolt. This action potential travels down the axon, and when it reaches the terminal bouton, it activates the voltage-gated calcium channels. When the voltage-gated calcium channels open, calcium can rush from an area of high to low, therefore calcium rushes into the terminal bouton. What is calcium good for? You may or may not know this, but this protein, this neurotransmitter that's stuck in the vesicle, the only way to get that out of the cell, because proteins are polar, they just can't pass through the cell membrane by themselves. The only way to get this out of the cell is through a process called exocytosis. So the movement of the vesicle to that plasma membrane, the fusion of that vesicle to that presynaptic membrane, and the release of the acetylcholine into the synaptic cleft, that's called exocytosis. And the biochemical marker, the biochemical molecule, in essence, that causes this exocytosis, it's calcium. So as the calcium concentration increases inside of the bouton, as more and more calcium rushes in here, it causes exocytosis, of those neurotransmitters. And now those neurotransmitters are in the synaptic cleft here, this space occurring at the synapse, and the acetylcholine is going to diffuse out and is going to get trapped by the motor endplate. When it does, it activates nicotinic receptors. Nicotinic receptors are non-specific monovalent cation channels. They allow sodium in faster than potassium can leave. Therefore, there's a net positive charge that builds up on the inside of that motor endplate, and it travels out to the sarcolemma and creates a new action potential because it activates voltage-gated sodium channels, and then we're going to repolarize and reset ourselves to norm by the voltage-gated potassium channels, and then action potential starts its next journey. That next journey is excitation, contraction, coupling, and that's for another video. Finally, I want to point out flowchart over here. Okay, flowchart. Simplify your lesson plan using flowcharts. Key words. What happens first? An action potential from the nervous system, from the somatic nervous system for the skeletal muscle, which activates the voltage-gated calcium channel found on the terminal bouton, drives calcium into the cell, reliant on the calcium concentration gradient, causing exocytosis of the acetylcholine out of those vesicles into the synaptic cleft, which is then going to activate nicotinic receptors, which are those monovalent cation channels, net sodium influx, which ends up making an endplate potential. Voila, neuromuscular junction. The last thing every single one of you guys should start to think about, if you're watching this video, it means that you care enough about anatomy and physiology to actually try to learn neuromuscular junction. And the only way to truly learn something is to break it. What would happen to a patient that has, right? Okay, a patient that is hyper-stimulated action potentials. More action potentials come down than normal. Nice, easy way to start to think about this. There's more action potentials than those voltage-gated calcium channels. More of them will be activated, and they'll be activated longer. More calcium influx, more exocytosis, more acetylcholine, more nicotinic receptors get activated, a greater endplate potential, more muscle stimulation. And that should only make sense, right? That's logical. So if I increase this with an arrow, if I increase this and say there's more action potentials, what's going to happen to the activation of voltage-gated calcium channels? That goes up. Therefore, what happens to the calcium influx? That goes up. Therefore, what happens to exocytosis? You guys get the point. What if we break something? What if, for whatever reason, we decrease the amount of acetylcholine in the synaptic cleft? Let's decrease the amount of exocytosis. It doesn't matter how you do it. What if you decrease the amount of acetylcholine out here? Let's say that decreases. Well, what's going to happen to everything downstream from that? That decreases, that decreases, that decreases. You're going to end up with less muscle contraction, right? Flowcharts are the key to studying people. Flowcharts make a very complicated subject so much easier to think about. Once you learn one flowchart, you learn everything about it. You can break this system a dozen different ways, and you can always answer that test question, that homework problem, that quiz question using the flowchart, because you're always going to be able to get to the root of the problem. That's the end of neuromuscular junction. Humankind, be both. Have a great day.